Hello, my name is Nir Hanania. I'm a postdoc in the Muir Lab. And I'm going to tell you today about my research. So one of the major subjects of research in the Muir Lab is chromatin. Chromatin is a complex of proteins and DNA found in the nucleus of eukaryotic cells. And its main function is to package the DNA, the very long thread of DNA, into the micron-sized nucleus. But what makes chromatin research so interesting is and important is its connection to epigenetics. The chemical structure and organization of chromatin dictate which genes are expressed in every cell, which basically define the cell identity. And you can also imagine that sometimes the regulation of chromatin goes wrong, so the cell undergoes some identity crisis, which can lead to cancer. So cells regulate or modify the structure of chromatin in large part by decorating the histone proteins with chemical modifications. For example, lysine methylation or acetylation. From a perspective of a chemist, studying chromatin is so exciting because you can really see the immediate connection between small, subtle changes in the structure of the proteins and the biological and functional effect. In the Muir Lab, we try to understand the biochemical function of different histone modifications. We explore how these modifications impact the structure of chromatin and how this chemical information is integrated and interpreted by the cell to elicit specific outcomes. Studying chromatin in cells is really hard because of its tremendous complexity. The approach that we often take in the Muir Lab is studying chemically defined facsimiles of chromatin in vitro. Specifically, we use our expertise in organic chemistry and in protein ligation technologies to make what we call designer chromatin. We make synthetic chromatin substrates in which we can control the chemical structure completely. We can install precisely any histone modification within our designer chromatin, and then we can study the biochemical functions of these histone modifications. Now, let me show you one of the instruments we use to make these chromatin reagents. So our designer histone proteins are semi-synthetic. We make them by ligating recombinant fragments that we make in bacteria and synthetic peptides. What you see here is a peptide synthesizer. It's a very cool instrument which saves us all the hard work of making peptides. Basically, what we need to provide the instrument is all the different amino acids and reagents. We just need to program the instrument very quickly, and within few hours, we can get a very long peptide ready to go. Thank you for listening, and I hope to see you here in the Muir Lab.